Regulation of gene function in prokaryotes and eukaryotes. Gene regulation, a label for the cellular processes that control the rate and manner of gene expression, a complex set of interactions between genes, RNA molecules, proteins, including transcription factors, and other components of the expression system, determine when and where specific genes are activated and the amount of protein or RNA product is produced. There are mechanisms of gene regulation. First is regulating the rate of transcription, which is the most economical method of regulation. Next is regulating the processing of RNA molecules, including producing more than one protein product from a single gene. Next is regulating the stability of mRNA molecules, and last, regulating the rate of translation. Gene expression is the process where genotypes coded in the genes are exhibited by the phenotypes of the individuals. The DNA is copied by the RNA and then synthesized into protein. The process of transcription, which is the synthesis of RNA from a DNA template, is where the regulation of gene expression is most likely to occur. There are four steps of translation. The first one is initiation, where the small subunit of the ribosome binds at the 5' end of the mRNA molecule and moves in three directions until it meets a start codon, or AUG. It then forms a complex with the large unit of the ribosome complex and an initiation tRNA molecule. The second one is the elongation, where the subsequent codons on the mRNA molecule determine which tRNA molecule linked to an amino acid binds to the mRNA. An enzyme, peptidyl transferase, links the amino acids together using peptide bonds. The process continues, producing a chain of amino acids as the ribosome moves along the mRNA molecule. Translation is terminated when the ribosomal complex reached one or more stop codons, UAA, UAG, and UGA. The ribosomal complex in eukaryotes is larger and more complicated than in prokaryotes. The last step in translation is the post-translation processing of the protein. In translation, the mature mRNA is used as a template to assemble a series of amino acids to produce a polypeptide with a specific amino acid sequence. The complex in the cytoplasm at which this occurs is called a ribosome, where the mixture of ribosomal proteins and RNA consists of a large subunit and a small subunit. There are also gene control regions. The first one is the start site, a start site for transcription. The second one is the promoter. Transcription factors bind to specific nucleotide sequences in the promoter region and assist in the binding of RNA polymerases. Let's go now to the regulation of gene function in prokaryotes. Next is the enhancers. Some transcription factors, also called activators, bind to regions called enhancers that increase the rate of transcription. These sites may be thousands of nucleotides from the coding sequences or within an intron. An intron are portions of the gene that do not code for amino acids and removed from the mRNA before translation. Some enhancers are conditional and only work in the presence of other factors as well as transcription factors. Last is the silencers. Some transcription factors called repressors bind to regions called silencers that depress the rate of transcription. The term gene expression is sometimes used to refer to the transcription phase alone. Operon model. It is a self-regulating series of genes that work in concert. In bacteria, genes are clustered into operons, gene clusters that encode the proteins necessary to perform coordinated function such as biosynthesis of a given amino acid. RNA that is transcribed from prokaryotic operons is polycistronic, which a term implying that multiple proteins are encoded in a single transcript. The activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins which affect its ability to recognize start sites. These accessory proteins can act both positively, which are called activators, and negatively, which are called repressors. An operon includes a special segment of genes that are regulators of the protein synthesis but do not code for protein, called the promoter and operator. These segments overlap, and their interaction determines whether the process will start and when it will stop. RNA polymerase must create RNA by moving along the chromosome and reading the genes in the process of transcription. Next is the termination. In prokaryotes, there are two ways in which transcription is terminated. The first one is the Rho-dependent termination, where the protein factor called Rho is responsible for disrupting the complex involving the template strand RNA polymerase and RNA molecule. The second one is the Rho-independent termination, where a loop forms at the end of the RNA molecule, causing it to detach itself. The last step is the processing. After transcription, the RNA molecule is processed in a number of ways. Introns are removed and the exons are spliced together to form a mature mRNA molecule consisting of a single protein coding sequence. RNA synthesis involves the normal base pairing rules, but the base thymine is replaced with the base uracil. A classic example of a catabolite-regulated operon is the lac operon, responsible for obtaining energy from beta-lactosides such as lactose. A classic example of an attenuated operon is the TRP operon, responsible for the biosynthesis of tryptophan. The lac operon model, probably the most studied and well-known operon. In bacteria, such as E. coli, three genes are part of an operon that code for three separate enzymes needed for the breakdown of lactose, which is a simple sugar. A regulatory gene, located before the operon, continually makes repressor proteins that bind with the operator and prohibit the function of RNA polymerase. The system therefore remains off until a flood of lactose molecules binds with all available repressors and prevents their attachment to the operator. When the operator is free, the production of the enzyme to break down lactose continues until enough of the lactose molecules are broken down to then release repressors to recombine with the operator to stop production of the enzymes. Two additional types of operons exist that operate in the same way except for the function of the operator. The next type of operon is the TRP operon. The TRP operon differs because the repressors is active only when bounded to a specific molecule. For the remainder of the time, it remains unbounded and inactive in the absence of that molecule. Finally, in a positive twist, activators are used by a third type of operon to bond directly with the DNA, which allows the RNA polymerase to work more efficiently. Absent the activators, RNA polymerase proceeds at a slow rate. Regulation of gene function in eukaryotes. Unlike prokaryotes, multiple gene regulating mechanisms operate in the nucleus before and after RNA transcription and in the cytoplasm both before and after translation. In eukaryotic cells, the ability to express biologically active proteins comes under regulation at several points. First is the chromatin structure, where the physical structure of the DNA, as it exists compacted into chromatin, can affect the ability of transcriptional regulatory proteins, which are termed transcription factors, and RNA polymerases to find access to specific genes and to activate transcription from them. The presence modifications of the histones and of CpG methylation most affect accessibility of the chromatin to RNA polymerases and transcription factors. Next is the epigenetic control. Epigenesis refers to changes in the pattern of gene expression that are not due to changes in the nucleotide composition of the genome. Literally, epi means on, thus, epigenetics means on the gene as opposed to by the gene. Next is the transcriptional initiation. This is the most important mode for control of eukaryotic gene expression. Specific factors that exert control include the strength of promoter elements within the DNA sequences of a given gene, the presence or absence of enhancer sequences, which enhance the activity of RNA polymerase at a given promoter by binding specific transcription factors, and the interaction between multiple activity proteins and inhibitor proteins. Next is the transcript processing and modification. Eukaryotic mRNAs must be capped and polyadenylated, and the introns must be accurately removed. Several genes have been identified that undergo tissue-specific patterns of alternative splicing, which generate biologically different proteins from the same gene. Next is the RNA transport. A fully processed mRNA must leave the nucleus in order to be translated into protein. Next is the transcript stability. Unlike prokaryotic mRNAs, whose half-lives are all in the range of 1 to 5 minutes, eukaryotic mRNAs can vary greatly in their stability. Certain unstable transcripts have sequences that are signals for rapid degradation. Next is the translational initiation. Since many mRNAs have multiple methionine codons, the ability of ribosomes to recognize and initiate synthesis from the correct AUG codon can affect the expression of a gene product. Several examples have emerged demonstrating that some eukaryotic proteins initiate at non-AUG codons. This phenomenon has been known to occur in E. coli for quite some time, but only recently has it been observed in eukaryotic mRNAs. Next is the small RNAs and control of transcript levels. Within the past several years, a new model of gene regulation has emerged that involves control exerted by small non-coding RNAs. This small RNA-mediated control can be exerted either at the level of the translatability of the mRNA, the stability of the mRNA, or via changes in chromatin structure. Next is the post-translational modifications. Common modifications include glycosylation, acetylation, fatty acylation, disulfide bond formations, and etc. Next is the protein transport. In order for proteins to be biologically active following translation and processing, they must be transported to their site of action. Last is the control of protein stability. Many proteins are rapidly degraded, whereas others are highly stable. Specific amino acid sequences in some proteins have been shown to bring about rapid degradation.